Today we will be going over donor unit testing for syphilis, Chagas disease, West Nile virus, and Zika virus. This presentation is by Lindsay Wilco, Brenna Williams, and Victoria Jones. Also, just a side note before we really get into this, I have a cold, a pretty bad cold, so if you just hear sniffling and everything throughout, I apologize. So for this presentation, our objectives will be list all the tests that are currently required for allogeneic blood donation, compare and contrast the screening test with the confirmatory test for the following: syphilis, Chagas disease, West Nile virus, and Zika virus, and explain pathogen reduction technology and pangeneric detection testing for platelets. Okay, starting with our required test for allogeneic blood donation, we got our ABO, which will be your standard forward and reverse typing for all blood types, and that will include any investigation of discrepancies that you may come across. And then for our RH, if that one comes across as negative, then you'll have to continue on to do a week D test for that donor. And then our antibody screen, that one is just there to help detect any underlying allo or autoantibodies the donor might have. If your antibody screen comes back positive, then you will have to then go on to perform an antibody identification panel. And then for our infectious disease testing, we will test for hepatitis B, hepatitis C, HIV 1-2, HTLV 1-2, syphilis, West Nile virus, Zika virus, and T. cruzi. T. cruzi is also known as Chagas disease, so you can call it either, whichever one you prefer. All right, now we will start talking about donor unit testing for syphilis. When you test for syphilis, you can test for two different markers, either the T. pallidum antibodies to antigens marker, or you can test for an RPR, which is a rapid plasma reagent markers. The methods for each of them are different. So for T. pallidum antibodies, you can either use a microhemagglutination method or an EIA, which is an enzyme immunoassay. For the RPR, you're either going to use a particle agglutination or a solid phase red cell adherence. And when testing for syphilis, it's just kind of like a nice thing to keep in mind, I guess, that it's like a fun fact, I guess, I don't know. The transmission of syphilis through blood products is actually extremely low, so it's not common that syphilis is transmitted through blood. You're going to find it more commonly transmitted through like other bodily fluids. And then positive donors are more likely to carry other STIs on top of syphilis. And it's more common among high school age donors. And there's going to be a high number of false positives as well as a high number of true positives. So, I mean, that's not really like a peace of mind that like there's a high number of false positives, because you also got a high number of true positives. So, yeah. All right, so our first screening test will be our rapid plasma reagent, or RPR, and it is a non-triple needle assay. So it is a non-specific screening test for syphilis, and how it works by detecting reagent antibodies to the cardiolipin antigens in the blood. So that's why it's like a non-specific test, because it's not detecting specifically syphilis antibodies or treponemal antibodies. And so you'll either get a reactive or a non-reactive, and reactive donors will have a current infection or they were successfully treated within the last 1 to 2 years. So that just kind of means like your body, so because it's not specific, it can't tell whether or not you still have those antibodies or not, but it detects, because it's detecting the cardiolipin antigens, it's saying you're reactive, it's giving like a positive result. Or you can be a non-reactive donor, so you are not infected, never have been, or you were successfully treated for an active syphilis disease, but it's been longer than that 1 to 2 years. And so the RPR is no longer detecting any of those cardiolipin antigens and saying, like, flagging that this person has syphilis or they might have syphilis, or they need a confirmatory test to actually decide if they really do have an active syphilis case. All right, our next test will be our enzyme immunoassay, or an EIA. This is going to be our confirmatory test, and it's a specific test for syphilis. So it is going to be our treponemal assay. That's what it's also known as. And it tests for antibodies to the syphilis antigen. So that's why it's more of like the confirmatory test than the RPR, which is non-specific. And this one, yeah, like it stated previously, it detects antibodies specific to the P. pallidum, which is the spirochete that is syphilis. And if a donor has been infected, the EIA will be positive whether or not they have been successfully treated. So the RPR kind of helps with once they have been successfully treated after 1 to 2 years, then they'll be non-reactive. And then at that point they won't have to have the confirmatory. So it's kind of nice and better to do the RPR before you do the EIA or the treponemal assay, because whether or not they still have syphilis, the treponemal will say they have it. So that in that case you could get a false positive. All right, so here's a nice little flow chart that I made for just kind of like to walk through syphilis testing. So we're going to start with our treponemal screening test, which will be our RPR. And either you're going to get a non-reactive or reactive. If the donor comes back as non-reactive, then they're good to go, good to donate, use their blood, be on their merry way. If they're reactive, then the donor will be deferred indefinitely. The donation may be okay, but because it's reactive, we're going to have to go on to more testing, do a confirmatory test, and whether or not the donation and the donors will both go through the confirmatory testing. And so our treponemal testing, which will be our EIA, and that will either come back as negative or positive. If it comes back as negative, then the donation may be used, or the donor will be cleared to donate or continue donating. If it comes back as positive, then that just means that the results are showing an active infection or treatment of a syphilis infection. Remember, like the EIAs, whether or not you've been successfully treated for syphilis, it will come back as positive regardless. At that point, if the donor or donation is positive, confirmatory positive, then the donor can not donate, and the donation is unusable at that point. All right, so say a donor tests positive for syphilis, but they still, down the road, they want to be able to donate, and they're wondering if they can. The answer is yes, they can, but they have to follow certain steps and eligibility requirements to be able to donate. And some of those are that the donor reports they were treated completely and successfully for syphilis at least 3 months before their donation. So, I mean, exactly like it says, they have to wait at least 3 months following the completion of a successful treatment for syphilis before they can even donate again. And then the donor also has to be determined by a physician that they haven't been infected with syphilis, or that they are no longer infected with syphilis through subsequent testing. And then on top of both of those, the donor must meet all of the eligibility requirements to donate. And if the donor does not meet the requirements or does not follow the steps to be cleared, then they are not eligible for re-entry to donate.
Hi everyone, my name is Lindsay Vanderwood and I will be taking over this next part of the presentation. So we're going to be discussing West Nile virus. The marker we're looking for is West Nile virus RNA. We don't want to look for antibodies on this one. The testing method, and only testing method, is nucleic acid. This can utilize transcription median application or PCR. So, moving on to symptoms of West Nile virus, most individuals are asymptomatic. However, some may show febrile illness. This can include fever, headaches, vomiting, body aches, and fatigue. Also, less than 1% of people infected develop a serious neurological illness. This can be encephalitis, which is generally the inflammation of the brain, or meningitis, which is inflammation around the brain or the spinal cord. The vector for this is mosquitoes, and this is generally the Chagas species. This is an arbovirus, which means that it is transmitted by arthropods. However, the natural reservoir for this virus is not in humans, it's in birds. So, mosquitoes will feed off the birds will start to die, so mosquitoes will move on to horses and humans, which are considered to be the end host or end of transmission. I'd also like to portray the prevalence of West Nile cases. So, showing in 2014, there were about 2,122 cases of West Nile reported by the CDC. Those who are most at risk for West Nile virus are generally the elderly or the very young, or anyone who is immunocompromised. So there's no vaccine or no specific medications available for West Nile virus, so it is important for these individuals to especially take precautions such as wearing bug spray or clothes that'll cover their skin. Alright, so let's get into the detection method. Most blood centers actually pool their samples together to screen for West Nile. Depending on the season, the size of those pools can vary. So in the summertime, there are smaller pools. That's generally because mosquitoes are out and about. And then in the winter, there is a much larger pool due to mosquitoes not being as prevalent. So if there's a positive within that pool being tested, then there will be individual testing that is performed afterwards. This saves the laboratory money and not taking the time on screening every individual person. But since West Nile is pretty uncommon, why are we screening for this in blood centers? The reason is it's screened by blood centers because many individuals are actually asymptomatic. Apparently it's about 80% according to the CDC. So those generally getting transfused are most likely to be sick, and we don't want the possibility of a transfused individual getting West Nile virus. Okay, so let's move on to a review of the transmission of West Nile virus. I personally am a visual learner, so this tends to help me a little bit. So we'll start off with the mosquito. The mosquito is the vector of West Nile virus. Do you guys remember the typical species that is? I can give you a second so you can have a minute to remember. All right, if you said the Chagas species, way to go, you're a champ, you got it. So the infected mosquito will feed on the blood of birds, and then the infected birds are considered to be the natural reservoir of West Nile virus. However, the birds will die from this virus, so the mosquito moves on to feed on the blood of horses and humans, and then they are too infected. However, they are considered to be the end of transmission or the end host. So just to review, it goes from the infected mosquito to the birds, which is the natural reservoir. The bird dies, moving on to the horses and humans, and that is the end of transmission. Moving on to the Zika virus. So Zika virus is the most recent test to be added to screening in blood centers. The marker would be Zika RNA, while the testing method is nucleic acid test. However, FDA approval is still pending on this, and it should only be used for investigational use only. All right, focusing on the symptoms of Zika virus. We want to focus on one main point. It causes birth defects. So this is commonly associated with microcephaly. For those that don't know what it is, it's when a baby's head is smaller compared to other babies of the same age and sex. This is especially important because these babies typically have an association with having smaller brains that are not developed properly. So it is important for pregnant women to implement prevention such as bug spray and wearing skin covering clothing. Next, studies have also found that there may be a link to Guillain-Barré syndrome, which is a person's own immune system damages nerve cells. And then some signs to look for when you are infected would be fever, rash, headache, and joint pain, and muscle pain and red eyes. So as we went over with pregnant women, prevention is key because the vector of Zika virus is mosquitoes, and typically this is the Aedes species, which will feed typically at day and night. So it's important to be careful. So blood centers can also pathogen reduce this product, with a method that is FDA approved. And if they are successful in the method, then they do not need to perform nucleic acid testing. This method is also called pathogen reduction. However, pathogen reducing is only available for platelets and plasma. So what is pathogen reduction? Pathogen reduction is a new technique that utilizes a substance called aminosaline, which can replace blood culture bottles. Bacterial contamination of platelets is a major cause of transfusion-related fatalities after trolley and hemolytic reactions. This is generally caused by bacteria on the donor's skin or a donor that has asymptomatic bacteremia. So pathogen reduction is a more effective way to test platelets for bacterial contamination compared to blood culture bottles. This is because blood culture bottles take too much time for bacteria to grow. So pathogen reduction has the ability to inactivate a broad spectrum of viruses, such as gram-positive or gram-negative bacteria, spirochetes, parasites, and blood T cells from the donor, which is associated with TAG-VHD prevention. Once the aminosaline is exposed to UVA light, it binds and is irreversibly blocking the replication of DNA and RNA, basically binding the nucleic strands together so they can't replicate. This only works on clear donor units, such as platelets and plasma. This is due to shadowing. An example is that the UVA light cannot penetrate a red blood cell. So this method is very important for donor testing, and I believe Brenna will be reiterating this test with a diagram, so I hope you guys pay attention, and I appreciate you taking the time to listen to my part of the presentation. Thank you.
Hey guys, Brenna here. I will be taking over the presentation. From here, I will be discussing Chagas disease, pathogen reduction, and pangenera detection. We're going to start off with Chagas disease. Chagas disease is one of the donor screening tests that we do on donor blood. Chagas disease is also known as Trypanosoma cruzi. This is a protozoan parasite that is found in your bloodstream. The testing was first recommended to do in 1989, but it wasn't until December of 2007 that the EIA, or the enzyme immunoassay test, was licensed by the FDA. There are two different testing methods that we use to test or screen for Chagas disease, and they are the chemiluminescent immunoassay, or the CHLIA test, and there's also the enzyme immunoassay, or the EIA test, that was mentioned before. There's also a confirmatory test that we use to confirm the test or the screening of Chagas disease, and that is the RIPA test, or the radial immunoprecipitation assay. So with Chagas disease, with the screening test, we only have to screen for it once. So the first time a blood donor comes in and donates blood, we will screen them for Chagas disease, but after that we won't have to screen them again. Once the donor comes in, we will get the blood and we will test for the IgG antibodies for Chagas disease. If it's negative, they won't be tested again, as I said before. The reason why we do this is because the testing is not that great of a test. But if the donor has tested positive on the screen, they will be permanently deferred regardless of the confirmatory test results. Only about 25% of people test positive on the confirmatory test. The vector for Chagas disease is the Thiamidin bug, or also known as the kissing bug. It will come onto somebody's skin and it will eat their blood, or drink their blood. And then after it does that, it will desiccate where it has eaten, causing the parasite to go into your bloodstream. Chagas disease is an endemic disease that is mostly in Mexico, Central America, and South America. It is estimated that over 11 million individuals are infected in those regions, but about 300,000 people have been tested and has tested positive for it here in North America. We will now continue on and discuss pathogen reduction. What is pathogen reduction, or what does it do? It inactivates a broad spectrum of viruses, gram-positive and gram-negative bacteria, spirochetes, parasites, and donor T cells. It also helps prevent the prevention of GVHD, or transfusion-associated graft versus host disease. This test is also FDA approved. This test is a technology that helps by replacing blood culturing, but this test only works for platelets and plasma. It does not work with RBCs. So how it works. So basically, it uses serolin, or amtololin, that intercalates into the DNA and RNA of pathogens and donor T cells. Once exposed to UV lights, amatosolin binds and irreversibly blocks to the replication of DNA and RNA. So let's draw a diagram of this. So we're going to draw the DNA strand. So we're going to do this. So this is the DNA strand. It's a terrible drawing. Excuse my drawing. And so now we're going to have the amatosolin, which will come in here and bind the strands together. And then we're going to have UV light come. And therefore, as you can see, that the things that are in blue is the amatosolin, and the red is the DNA strand. So since the UV light has come onto the amatosolin, it binds and it won't break or anything. Therefore, it won't have the DNA unravel to be able to duplicate or replicate for that disease or the bacteria and everything. So, which is pretty cool, I think. So, we're going to go on to pan-genera detection. This detection is also used on platelets. It's another test that we use for platelets. It's also known as a rapid test. It has been FDA approved as an adjunct QC test for leukoreduced platelets. It detects the lipopolysaccharides in gram-negative bacteria and the lipotoic acid in gram-positive bacteria. To be able to use this test, we use what is called the sandwich immunoassay. Basically, that assay has antibodies of a particular organism that is embedded at the bottom of the well of a plate or something. This type of immunoassay helps us prevent bacterial contamination. Thank you for tuning into my section of the presentation. I hope this was helpful for you, and continue watching the presentation. Thank you.